What we do is, when the biologists have some problems or questions to deal with the chemistry part, when they want to generate models or when they want to generate hypotheses, they come and talk to us to help out with generating a model. And sometimes it can be just a mere idea. We are trying to use our molecular modeling tools to help out with their experiments, or sometimes to come up with hypotheses, or sometimes to verify certain aspects of their experiments.
If you really want to understand what's going on, you need to understand the energetics, the contributions of the residues that might contribute to disease, or how they contribute to how the protein functions at an enzymatic level, and this requires computational chemistry. You really need to be able to go in and understand the energetics and how each one of those residues contributes.
We will be able to, say, get the structure of the allergen from peanuts using our empirical techniques, and then we want to expand that into looking at related allergens, maybe from peaches, maybe from birch pollen, some other things. And so what we do is we ask Lalith to start modeling the interactions for these closely related allergens to help us expand what we've been working on.
What we do is get that information and feed it to our molecular modeling packages, and then give an environment which is somewhat representing the physiological environments that you can see around cells. We use this physics-based methods to run molecular dynamics type simulations. We are partly using the data from your structures and then create more data to visualize what's going on.
You can kind of construct and deconstruct the protein to give us an idea of how each part kind of interplays with the whole, and give us better ideas about the dynamics of proteins and give ideas for how to guide lab work in the future.
There are two types of tools that we are using. The first type is the quantum chemistry tools. Assume that you are dealing your molecules in atomic level, deciding what's going on within your reactions. We have to use quantum chemistry tools, basically the programs that are written to calculate the electronic structure of the molecules that you are interested. The second type is the molecular modeling tools. Suppose you have a big protein. There you have to use a little bit more coarse graining, and atoms are represented by charge centers, and then we let physics take care of how they interact.
We use different files to map kind of the topology of the protein and give an idea of what forces are going to be at play to give us an output of the dynamics. It's basically imposing the physics of the system on the topology of the file to give us the overall protein dynamics.
Being able to interact with Lalith is extremely valuable because what we try to do is go into the lab and make some mutations, test them, and then come back to him with ideas for what does he think happened when we made that. He's excited about trying all of the different novel techniques that computational chemistry has to offer, and he's always willing to see what happens and accept feedback for what he's doing and how it can be related to our project and what we're working on.
The Computational Chemistry Core here at NIEHS is the only group with the expertise to carry out the types of studies that they do, and because their work is so applicable to so many of us, it's really a critical part of the infrastructure we have here.
For me, it's like solving puzzles. Every day, if I can solve a puzzle, then I can get more excited about solving more puzzles.